transcription. Okay. All right. So as people trickle in, I'll start by introducing our speakers. So our speaker today is Dr. Aparna Bhaduri. She got her degrees from Rice, same year as me, 2010, a good year. And then she was at Stanford for her PhD, focused on epithelial tissue differentiation and neoplasms. She then moved on to UCSF, where she did a postdoc with Dr. Arnold Kriegstein. I guess her bio is a little bit more on the science, but I'll actually give her a chance to introduce all that good stuff. And so she started her lab at UCLA in 2021. And yeah, I'm looking forward to hearing about this exciting fusion of neuroscience and cancer and spatial biology. So take it away.
Thank you so much. Really appreciate that introduction. And let me try to see if I can revisit my sharing. And maybe you can just confirm when you see it, if you can see the slides. You good?
Yeah, I can see it.
Cool. I can't see any of you because I don't really know how to use Teams, so I'll just go, but interrupt me if anything comes up, and I'm happy to take questions along the way. I really appreciate this introduction and invitation, and as I was telling Alex, this isn't exactly a spatial, I'm sorry, let me see if I can close my email because that's not helpful. This isn't really let me make sure I'm sharing again. Sorry, I'm not a Teams person, I apologize. This isn't really spatial sequencing or spatial transcriptomics, but really we are very interested in understanding the spatial context and the spatial interactions of glioblastoma. So I would say for those of you who are looking for more of a technology talk, I'm sorry if I disappoint, but I do think that it's really interesting to think about the ways in which we can talk and think about glioblastoma and to think about these physical interactions. And so that's why I've titled the talk Understanding the Context of Glioblastoma, and I hope that it will still be interesting even though there's limited spatial analysis specifically. Also, this is the first time that I'm getting to present some of this data from my lab, so I'm really excited and I'm happy to have conversations and discussion. What you're looking at here is an example of the tumor transplantation that we do in what we call our HOT system. And in this system, we take glioblastoma cells, which are a very aggressive tumor, and I'll go into all of that background. But what you can see here is how they are growing in a cortical organoid. And so these green cells with this beautiful morphology shows us some of the heterogeneity that exists in these tumor samples. And I think that it just really gives us a visual understanding of how we can use this model to understand the tumor and also a lot of the complexity of the tumor. So as a little bit of an introduction to my lab and kind of our trajectory, we are really interested in understanding neural stem cell dynamics and thinking about the ways in which neural stem cells function in normal development and in glioblastoma. So we know that neural stem cells, often referred to as radial glia in the cortex, are able to give rise to a huge diversity of cell types that exist in the adult human brain. And when we look at this trajectory, we see that there's a lot of different cell types, different trajectories, different processes. And one of the best ways that we can really understand what's happening is to profile actual developing samples from the developing human brain. And we've been doing this as part of, I did this as a postdoc, and now my lab also is contributing to these efforts as part of the BRAIN Initiative. On the converse, we're also really interested in looking at those same dynamics because many of these cell types and trajectories are reactivated in aggressive brain tumors, including glioblastoma. And so again, from primary samples, we're really interested in characterizing what cell types and trajectories exist. But in both cases, whether it's the developing brain or it's in glioblastoma, primary tissue alone can only get us so far. And we need systems where we can manipulate the system in order to really understand what's happening. And that's where these cortical organoids come into play. And they really allow us to ask questions about these neural dynamics in development, and then also to think about this in the context of glioblastoma through that transplantation method that I mentioned. With these systems, we're doing comparisons and manipulations across systems and really thinking about cell type heterogeneity, gene expression, and then other modalities that we're also measuring to understand the underlying biology of what's going on. So the big questions that my lab is trying to ask are, how do human neural stem cells build the human brain? And why are they so devastating in brain cancer? And so many of you probably are aware of glioblastoma, but it's the most common and most aggressive form of adult brain cancer. And one of the things that's really problematic about it is that these tumors just almost universally recur, and they're highly heterogeneous, which we think may be one of the things that makes them so hard to treat. Also, we have seen that these tumors have many characteristics of development, and this has been shown by a number of people dating back to the first molecular characterizations of the tumor and continued through single cell profiling. And we ourselves have done some of these comparisons and have noted the reactivation of specific cell types. And so really thinking about this, glioblastoma is an adult tumor, but it really is also a developmental question. Now, some of the work that I did as a postdoc was as part of this brain initiative, and we performed single-cell RNA sequencing. I'm pretty sure that this audience is very familiar with looking at single-cell plots, but for anyone who is not, these types of UMAP plots have individual dots representing a cell, and they can be colored by any type of category that we've used to analyze these samples. In this case, this is from the second trimester of human brain development, and they are colored by the major cell type or state for the individual populations. So excitatory neurons, inhibitory neurons, dividing cells, radial glia, etc. And as a postdoc, we were really interested in looking at these cell types through the second trimester of development, which is the stage of peak neurogenesis. And when we had these atlas scale data sets, we were able to then start comparing them to glioblastoma. And in doing so, we identified the activation or reactivation of outer radial glia, which are a unique subtype of radial glia, a progenitor cell in the brain that showed us that there was reactivation of developmental cell types. And I'll do a very brief vignette on this before building on it further. But when we also looked at the other cell types that existed in the tumors, we saw that there were many other cell types of interest as well. And a number of these also had developmental features, including in astrocyte populations and oligodendrocyte populations, suggesting that this might be a more overarching principle in glioblastoma as it relates to individual cell types, which hadn't been quite as well elucidated at the time that we were doing this. So when we think about the reactivation of outer radial glia, one of the ways that we looked at this was to compare subtypes of outer radial glia, truncated radial glia, and ventricular radial glia that exist in development to the radial glia populations we were seeing in glioblastoma. And we saw that the highest correspondence was to this outer radial glia population. Now, outer radial glia are expanded in the human brain compared to primates, and compared to rodents, they're very sparse in the rodent brain. So this suggests that this is a human cell type that is enriched in human development that is coming back in glioblastoma. And this is really interesting to us because even in context of like neurodevelopmental disorders, the things that are most evolutionarily recent or expanded are the things that are the biggest vulnerability for disease. And so this was one thing that was really interesting to us. And we saw that the gene expression network was well preserved. So this is a network of genes that exist in outer radial glia during development and that are reactivated during glioblastoma. And one of the genes that's at the center of this is this gene called PTPRZ1, which is a receptor tyrosine phosphatase, and it'll keep coming back in this talk, so I just wanted to highlight it here. Now, one of the things that's really interesting about outer radial glia is not only were we able to see their transcriptional signature reactivated in tumors, but we were also able to see a unique behavior, this jump and then divide that I'm showing in this video here. And this is a behavior that has really been associated with the cell type and hasn't been seen outside of the developmental context. And what we observed is not only was there this jump and divide, but also after that jump and divide, the daughter cells that emerged within tumors, so these are sparsely labeled tumors for which we did live imaging, also were proliferative, suggesting that this was a population that is really generating proliferative daughter cells and thereby propagating the tumor. You can see these divisions here of those daughter cells. So this told us that there's some very important role for the cell type, But one of the challenges that we were having is that we were unable to necessarily isolate the population and then grow it in a in vivo environment. And while this might be a gold standard for determining whether or not something is a tumor, stem cell, or a progenitor, we thought that the fact that this cell type exists and is reactivated after decades, when we think it disappears by birth in the human and it comes back 6 to 7 decades later, indicates that we really should explore what it is doing. And so we started by using, again, PTPRZ1 because it's a receptor, to isolate these populations and to transplant them into cortical organoids. Now, for those of you who are not familiar, what is a cortical organoid? So a cortical organoid is a stem cell-derived model of the developing human brain. And the way that we make them is from pluripotent stem cells. These can either be human embryonic stem cells or induced pluripotent stem cells. We're then able to aggregate them in the presence of small molecules, which allow us to make these small balls and direct them towards the cortical fate. And then over time, they grow up significantly. When we basically cut them open, fix, section, and stain them, we can see that there are these rosettes that are the progenitors, or the radial glia, marked by SOX2, that give rise to intermediate progenitors, or transit amplifying cells within the organoid and the developing human brain, and then markers of deep layer neurons, such as CTIP2. So we can recapitulate some of the major cell types that exist in the developing human brain, and this is a really unique tool because it allows us to have a human cell microenvironment as a substrate for the tumors that we grow. And I'll talk a little bit more about our characterization of these cell types later. In the previous work that I did, we did see that enriching radial glia was not a perfect enrichment, but it did allow for pretty good depletion of the negative fraction. And across the sorted, unsorted, and negative sorted populations, we were able to derive heterogeneity of cell types after several weeks in culture, suggesting that indeed outer radial glia cells are capable of giving rise to additional populations within the tumor. So when I started the lab, I wanted to build on this method, and Dr. Wei-Hong Ji is a very talented associate project scientist in the lab now, and she really took on the job of optimizing this system. And when we had done the work in the previous paper, it worked, so we kind of used standard conditions, and we thought, you know, hey, let's just use the system because it worked. But when I was starting my lab, I thought, you know, maybe we should optimize this a little bit more, figure out if there are better conditions, if there are ways of making the system have more fidelity to the patient brain. And so what Wei-Hong spearheaded was a project where we looked at 5 different tumors, and these came either directly from patient tumors, which is in the case of 3 of them, or 2 that came from xenografted tumors that had never seen a culture dish and had gone brain to brain. These were in collaboration with Dr. David Nathanson here at UCLA. We then dissociated the tumors, and we kept some for either single cell or single nuclei RNA sequencing, and the rest were dissociated into single cells and labeled with GFP. Wei-Hong then tried a number of different methods to do the delivery of the tumor cells to the organoid. I had a method that I had devised as a postdoc, and it was good, but not great. And so Wei-Hong made it better. And essentially what she did is she created these droplets where she's able to put a trop of tumor cells around a cortical organoid and then leave it in a hanging drop system for 12 hours. This really drives the tumor cells inwards and allows them to invade the system. And you can kind of see what that looks like, where at day 1, they're really on the periphery. After a week, the edge of the organoid is somewhere here, and you can see that they're going in. And then by week 3, they've really made like little independent balls in some places, and then other places you can see the invasion. And this is similar to what I showed on my first slide. So the delivery was one thing that needed optimization, and she really optimized the cell number as well. But then she also wanted to look at how different media conditions may or may not affect the environment. Does the media in which these organoids are grown impact the ways in which the tumor cells are able to function? And so we had 3 to 4 weeks in 5 different media conditions, after which we sorted out the tumor cells and performed single-cell RNA sequencing. And then we used some RNA scope validation as well as immunofluorescence to really characterize the cell populations and their distribution spatially within the organoid. Now, I'm going to take a quick detour because one of the ways in which we annotated cell types in the single-cell RNA sequencing was through a lot of our analyses, was through a meta-atlas approach. Essentially, the idea being that because there are so many data sets that exist both for development as well as for glioblastoma, if we can put them together, we can get a better consensus of the cell types that exist in the glioblastoma as well as in the developing brain. So this is a method that was innovated by Patricia Nano, who is a postdoc in the lab, as well as Elisa Fazzari, who is a very talented MD PhD student. And this method in development is on bioRxiv, and that's Patricia's work. So check it out if you're interested. But the general gist of what this approach is that we're taking published, in this case, glioblastoma single-cell sequencing data, and looking at each tumor individually. The goal here is to be able to find gene networks that are describing biological processes that we can then correlate back to understanding cell type. And it allows us to look and overcome some of the technical or patient-specific variation that is inherent in these tumor analyses. And so what Elisa did in this case was perform tumor-specific unsupervised clustering, where she identified tumor-specific gene expression signatures, aka the genes that are most variable and highly correlated within each individual tumor, that she was then able to compare across the individual data sets to integrate the expression and then to generate a series of networks that we used to annotate cell types across the glioblastoma samples. So the inclusion criteria were published primary GBM samples. In this case, we only looked at adult and only at IDH wild type. Additionally, we really wanted to focus on tumor programs because we're really interested in this study for the tumor biology and not necessarily yet looking at the normal microenvironment or normal cells that are captured during the process of sequencing the tumor. So many of the studies had already done copy number variation, and for those that hadn't, Elisa identified which were the tumor cells. So in the end, we wound up with about 73,000 cells across 55 tumors and 7 data sets named here. We then were able to basically take these networks, and this is what that last step looks like, where we do some of these gene correlations to identify networks within the data set. And for the purpose of glioblastoma, Elisa identified 152 metamodules that ranged in size from 10 to 323 genes. And these are groups of genes that we think are biological processes identified by multiple data sets that then allow us to really think about the cell types that are represented in the tumor. So if we visualize this, and I want to highlight that the visualization is independent of any of the meta module creation, but it's a way for us to orthogonally look at the data in a way that is pleasing. We can see that there are many clusters that are generated, as well as an integration of data sets based on the UMAP space for visualization. We then leveraged a number of different tools, including the clustering as well as the meta modules, to identify the major populations, and we're able to find 14 to 15 cell types that we're seeing within the tumor. And then if we correspond this to some of the published data that exists in the field about major states that exist, we can see that there is coherence of these states. So apologies for the digression, but basically this strategy for GBM was where we really anchored our cell type annotations, because it gives us an opportunity in this model that we call the human organoid tumor transplantation HOT. It gives us an opportunity to link this back to kind of coherent primary cell types that we can then use to compare between the transplanted cells across conditions and the primary tumor. And so this is a integration of the meta-atlas and our single-cell sequencing from the HOT system across media conditions. And this is just the HOT samples as they are projected onto the system. And you can see that most of the populations are well represented, although some of the more mature populations like oligodendrocytes are not preserved in this system.
Hey, Aparna?
Yes.
Real quick, I don't know if you've been pointing with your mouse, but I think you'll need to turn on the laser pointer. At least I can't see your mouse when you're referring to specific panels.
Got it. Now can you see it?
Yes.
Okay, thank you for that. I appreciate it. Great. So one of the things that we wanted to look at is we had used 5 different media conditions. These included our baseline conditions, which is our media 3, which is one of the medias in the organoid context. We had then used a version of this without FBS, and this is really important for glioblastoma culture because gliomospheres and other tumor cells typically react pretty badly to FBS, and so we wanted to make sure that we had a condition that accounted for that. We also wanted to look at what happens if we add in growth factors. This is typically the type of culture condition that is used for gliomospheres or other types of line cultures. And then also another version with PDGF, where we wanted to see if we could specifically promote oligodendrocyte or oligodendrocyte precursor cell development in the system. And then we also had a version where we added B27 to see if this would change some of the neuronal composition. And what we looked at was that although there were some differences in proportions of which cell types existed, the heterogeneity within a tumor across the different media conditions was relatively stable. So if you are getting neuronal cells, outer radial glia, and cycling cells, you're still seeing that across the different media conditions. Now, there are a couple cases where the FBS-free conditions failed to implant, and so this is something that we wanted to consider in terms of deciding which is the best condition moving forward. And we also wanted to quantify the number of cells, and so although that's not shown here, we did see that the largest number of cells were either in this M3 no FBS plus growth factor condition or in the media 3 condition. Now, one thing that was really interesting to us is we have done a number of characterizations of the system kind of at baseline conditions. And again, you can see here is an outline of the organoid. This is what it looks like when the system is live. And then there's invasion of primary GBM cells into the organoid. And when we fix section and stain this, we can see that these GFP positive cells are maintaining Ki67 expression, nestin, and SOX2. And nestin and SOX2 are some of the key markers for stemness. And this was really interesting to us because this particular example is shown in the FBS condition and suggests that although these cells, when exposed to FBS just without a microenvironment, will completely differentiate, in this context, we were seeing that those effects of pro-differentiation from the FBS were mitigated by the microenvironment. And the reason I want to highlight this is this was really one of the first ways in which we saw that you can maintain stemness in FBS conditions, and that this suggests that the microenvironment is doing something to change the way that the tumor cells experience their surroundings. This is just another example of these stemness markers with nestin, GFP, Ki67, and then SOX2 as well. And what we can see is that there is signal for these co-localizations, and we've quantified this, and we can see that it's true across conditions, but this is true across the different media conditions. So we're able to see these both yellow and in some cases white cells across the different media conditions, suggesting that stemness is persisting across the culture conditions, contrary to what we might expect. The next thing that we wanted to do is really to evaluate how much fidelity each of these tumor cells have to their primary tumor, as well as between conditions, i.e., we're making a system where we're able to preserve a lot of the heterogeneity. The cell types are there. But if I'm seeing a astrocyte in one media condition or in the primary tumor, is that the same astrocyte I'm seeing in a different media condition in the hot system? And so we did this in a number of different ways. I'll show you 2 of the analyses here. One of the ways that we did this is we used single sample gene set enrichment analysis, or SSGSEA, to look at the signatures for each of the cell types identified in the meta-atlas that were represented in the hot system. And from this, we were able to look at the distribution of the cells and how they score for each of these programs. And the key takeaway here is that overall, you're seeing that these 5 ridge plots are really overlapping. In other words, there's not major differences in terms of the degree of representation of each of these programs across media conditions. There are a couple examples, for example, here where there's higher representation of this vascular program or this neuronal program, but otherwise the distributions are highly comparable. This was specifically looking between media conditions. And then we can also look where we compare the primary tumors with either single cell or single nuclei analysis to the different media conditions. And what we can see is, although there is a variability based on the dot size, which is the number of cells that are retrieved, if you look at the coloring, for the most part, they're quite similar between the different media conditions and the primary tumors, with a few examples of a media condition lacking signal for an individual cell type. Otherwise, they're relatively conserved. And again, it's just the proportions that are changing. So this gave us confidence that the cell types that we're looking at in the hot system really are the same cell types that are existing in the patient tumor, sometimes with different mixes of numbers or proportions. Now, the next thing that we wanted to do was to understand how this system of organoids is supporting tumor growth. I think I've mentioned a number of times now that the tumors we are looking at are adult tumors. They're coming from patients who are typically in their 60s and 70s, sometimes a little bit younger, sometimes a little bit older, but they're not developmental contexts in which these tumors are grown. By contrast, the organoids that we are studying and that we are using as a substrate to explore the system are developmental models. They are intending to model developmental cell types and are primarily used for the study of human cortical development. And so we wanted to really interrogate how and why does this system work? And does it have anything to do with reality? So the first thing that we did is we had another meta-atlas, the one I mentioned that is led by Patricia, which characterizes cell types in the developing human brain. And this is during peak stages of neurogenesis, similar to the atlas that I explained to you from my postdoctoral work. And that data set is one of several that is included in that study. When we look at the predicted score of using a projection algorithm of the organoid cells to the developing brain, we can see that the vast majority have a score of 1, which is essentially a perfect match, and almost everything has a score above 0.5, suggesting that we have really strong enrichment for the characterization of cell types of the organoid as defined by cell types in the actual primary developing human brain. This makes sense. This is what we'd expect. It's a developmental system. Now, one thing that was really fascinating to us then is, as I mentioned before, when we do single cell capture of primary tumors, this is both from our own data as well as from other published data sets, we do capture some of the non-tumoral cells. So these are the cells that are in the peritumor environment or in the microenvironment. And we again did a comparison of these peritumor non-neoplastic cells to the developing brain, to the healthy adult brain, and to the aging brain. And we were shocked to see that the highest correspondence for these peritumor cells was actually to the developing brain, suggesting that there's something about the communication of the tumor and the microenvironment that is changing the microenvironment and making it look more developmental. This is also really interesting because it suggests that this is why the hot system works, that the microenvironment actually is not that different from the peritumor environment in the patient. So with this in hand, we wanted to kind of see what is this crosstalk that is happening in this context? What are the tumor cells saying to the microenvironment and what are they saying back? So the first way that we looked at this was to compare the transplanted organoids, the ones that received tumor cells, to batch-matched tumor-naive organoids. And just looking in UMAP space, we saw that there was some segregation of the populations between these two conditions, suggesting that there are some differences based on communication that are occurring. And if we look at the actual cell types that are represented, we do see a change in the proportion of neuronal cell types in the transplanted versus the naive condition. We aren't as interested in this particular study to think about what is happening to the organoid, because although I've just spent some time explaining to you why it's a justified model for the peritumor environment, we're not sure that these effects are necessarily as tumor relevant. But we did use this piece of data as evidence and motivation to use this system to study tumor microenvironmental communication. And in each of those cases, we want to anchor it back to actual patient data to understand what's happening. So we used an algorithm called CellChat, which looks at receptor ligand pairs within the data. and saw that there was a top communication pathway suggesting that there's communication between the tumor and the microenvironment through PTN PTPRZ1. Now, you may remember PTPRZ1 as one of the markers that we used to study radial glia in the system. And one of the things that we saw here is if we looked at one of the transplanted primary tumors and which communication pathway is mediating communication with the organoid, PTN was the ligand that came up as the top one. And if we look at its proportion in terms of its receptor, PTPRZ1 is the top. Now, previously we had used PTPRZ1 as a marker for outer radial glia, and you can see that it's very highly expressed here in terms of tumor outer radial glia. However, as you also saw in that plot, when we did the enrichment, it was only about a 40% enrichment, and that's because PTPRZ1 is expressed by other cell types in the tumor. It's the highest in the outer radial glia. So this was in the organoid system, and we wanted to make sure that this isn't an artifact of the organoid system. So we also did the analysis in primary cells, and we did this. I'm showing one data set here, but for every data set for which we had both tumor and non-neoplastic cells, we did this comparison and came up with the same answer, with PTN and PTPRZ1 signaling at the top of the pecking order. We thought, okay, this is really interesting because it suggests that there's both PTN and PTPRZ1 in the microenvironment, which is not what we thought we would expect from the healthy adult brain. So we wanted to confirm this, and we again looked at the healthy adult brain, and this is a data set from the Allen Institute, where we saw that there was substantially less interaction. Now, I know that this bar is still the top one, and we're thinking about ways of representing this data to explain this, to show this relative contribution. But if you look at the axes here, we're at 4 and 4 here, and here we're well below 1. And so you can see that the strength of these interactions is minuscule, and PTN is not at the top of the list here. Also, the number of interactions between cell types is much lower, and led us to conclude that this is not a common interaction that exists in the healthy adult brain. So presumably, because that microenvironment is different in the tumor compared to the healthy adult, we are seeing upregulation of PTN and PTPRZ1. Now, this is just what the data was suggesting. We wanted to validate this. This is the closest I'm going to come to an actual spatial experiment. I need to tell you a bit about PTPRZ1, but then I'll show you the pseudospatial experiment. So PTPRZ1, as I mentioned before, is a receptor tyrosine phosphatase. This means that it removes phosphate groups from downstream signals, often ones that are involved in cancer. And this catalytic activity is inhibited by its ligand PTN. And when PTN and other ligands bind PTPRZ1, and this is beautiful work shown by Michel Mongi in a number of in vivo and in vitro systems, we can see that there is a activation of the Rho-ROCK pathway because PTPRZ1 can no longer dephosphorylate this. And this pathway seems to be important for cell migration and invasion. Now, this was really interesting to us when we were working on my postdoc study on outer radial glia, because this Rho-ROCK pathway has also been implicated in that jump and divide behavior that I showed you earlier. And so we knocked down PTPRZ1, and we showed that this gene is required for that jump and divide. And this is true not only in tumor cells, but also in the developing human brain. So this is a link between the Rho-ROCK pathway, the mitotic somal translocation, and PTPRZ1. Now, we also saw this when we did an inhibitor of the ROCK pathway, where we were able to show that there's a decrease in a two-dimensional migration assay, and that this is phenocopied by PTPRZ1 knockdown, suggesting that PTPRZ1 does have a strong role in tumor migration, and that this is an important feature in a tumor intrinsic manner. Now, one thing that is really confusing is, as I mentioned, we're seeing this pro-cancer effect when PTPRZ1 is bound by its ligand, which results in inhibition of its catalytic activity. However, if you use a small molecule against PTPRZ1 to also inhibit its catalytic activity, this inhibits growth. So this suggests that there's a little bit of a contradiction here where different mechanisms of catalytic inhibition result in opposite growth and also migration phenotypes, which this paper did show, but I'm not highlighting here. Keep this in mind as we think about PTPRZ1 as a tool for communicating with the microenvironment, because all of these studies are thinking about the cell intrinsic role of PTPRZ1, but our data and preliminary data is suggesting that there's actually a communication role of a cell extrinsic or a cell non-autonomous role for PTPRZ1. So if this is all true, we need to see that PTPRZ1 actually exists in the tumor microenvironment, as opposed to just in our single cell data. So that's why we did this experiment. And this was in collaboration with Kunal Patel, who is a very talented neurosurgeon here, and who has been doing these really interesting resections based on some specific imaging tools. He's able to sample from the tumor core as well as from the tumor periphery. And obviously he's resecting the whole tumor core, and then he's doing some clinical trials where he's thinking about which parts of the periphery based on this imaging to resect. So given this, we were able to obtain a number of samples where we had a boundary between the tumor and non-tumor cells. And we could see this by two ways. One was through Neston staining as well as through the cell density. And so in these samples, we wanted to look to see, is PTN and PTPRZ1 expressed in the tumor core, in the tumor cells? And then is it expressed in the non-neoplastic cells that exist both in the core and the periphery? And so we were able to do this with 5 samples, and this was done with RNAscope. And you can see from these different insets that we have 1 is really in the center of the tumor core, 2 is really at the boundary, and 3 is in more of the periphery. And when we quantify this, we can see that there is expression of PTN and PTPRZ1 in both neoplastic and non-neoplastic cells in both the core and the periphery of the tumor. So this suggests that our model could be feasible in that this idea of an upregulation of PTPRZ1 not only in the tumor, but also in the microenvironment does exist, and it could be something that is mediating communication. So given that was the case, we thought, well, we actually have a great system to test this because it's incredibly challenging to perform microenvironmental manipulation experiments in other systems. You can't do that in the human brain. It's difficult to do in slice culture. And if you're going to do it in a mouse, you need some sort of complex genetic tools. So our HOT system really uniquely enables us to do this. So we wanted to ask some functional questions about PTPRZ1 biology. Ryan Can, who's a very talented graduate student in the lab, devised a migration assay using the HOT system. Now, I showed you previously that PTPRZ1 seemed to have an impact on migration. Some of that work was done in Michelle Mongi's lab at Stanford in an in vivo system, and then we, when I was a postdoc, had done some of that in a 2-dimensional system. But Ryan devised this 3-dimensional system that allowed us to do this in the context of the HOT system. And what he does is he has his transplantation of tumor cells. And in this case, we're using glioma spheres because we can just get really big numbers of those. And so it makes it quite amenable to this experiment. And then he fuses the transplanted organoid to a blank organoid. You can see that out here. And then 12 days, 18 days, and 25 days post fusion, you can see that there's this migratory front that's kind of going through into the blank organoid, which allows us to start to look at how much migration is happening. So we wanted to evaluate this migration in terms of PTPRZ1. And so to do this, we performed knockdown either of the tumor. So we had scrambled for tumor and organoid, knockdown for tumor and organoid, and then we did a mix and match. So there's 4 conditions where we can do scrambled tumor and knockdown organoid, etc. Ryan validated that we were getting knockdown both by Western blot and by qPCR. And importantly, in this case, our hairpin against PTPRZ1 is not targeting PTN. So we think that the results we're seeing are PTPRZ1 specific. He then quantified the migration a couple different ways. One of the ways that he did this was by using a immunofluorescence, where he was able to define the boundary from his sectioning, and we had larger tile scans that would really allow us to define the boundary between the two organoids, and then quantify the number of migratory cells. One thing that was really interesting here is when we looked at the number of migrating cells between the scrambled condition, which is going to be this purple condition here, and the tumor knockdown, we saw that there was a reduction in migration. This is consistent with what we were seeing previously, what's been reported previously. But here's where things start to get a little bit crazy. When we knocked down PTPRZ1 in the microenvironment, we actually increased migration, both on its own and in the context of tumor knockdown, essentially suggesting that environmental PTPRZ1 is a brake on tumor migration, but the intrinsic findings that have been reported previously still hold true in this system. Ryan also did this quantification numerically through FACS sorting, where he severed the organoids at the time of harvest and then sorted and counted the number of GFP positive cells on the transplanted side, on the fused side, and was able to quantify migration and came up with the same results. Again, showing that PTPRZ1 knockdown in the tumor does inhibit migration, but if you knock it down in the microenvironment, you increase migration, suggesting an antagonistic role between tumor and microenvironmental PTPRZ1. Now, because of the link that we had made previously between PTPRZ1 and outer radial glia, we really wanted to also look at whether this had any impacts on cell fate. Outer radial glia serve as neural stem cells within the cortex and therefore have an important role in generating other cell types. And we saw that through our videos that there's actual driving of creation of populations within the tumor. So Ryan did a number of stainings where, again, he was looking at 4 conditions. The tumor scrambled, the scrambled, the knockdown of the organoid, the knockdown of the tumor, and then the double knockdown. And in each case, he looked at whether organoid knockdown impacted tumor cell fate and was able to see for a number of markers, and here are the tumor quantifications for HMOX1 and MEF2C, that knockdown of PTPRZ1 in the microenvironment was changing the cell types in the tumor. Again, suggesting that there's some sort of a communication role. This was also true for GFAP and CTIP2, an astrocyte marker and a deep layer neuronal marker. And we saw some interesting spatial patterns where, especially when we did some of the manipulations in the tumor, that the organoid also changed in terms of its CTIP2, specifically in areas that were directly peripheral to the transplantation. So we really do think that there's a lot of crosstalk happening and that PTPRZ1 may be mediating some of this crosstalk. Now, as I mentioned before, there is a small molecule that will allow us to inhibit the catalytic activity of PTPRZ1. And so we wanted to see if we could use this small molecule in the system to define whether or not the role of PTPRZ1 in this cell-cell communication is in a catalytically dependent or independent manner. So this small molecule, NAZ2329, inhibits its phosphatase catalytic activity. So we can see that the molecule is working by an accumulation of phosphorylation shown here, which is quantified here. When we then look at the cell fate changes with this, we're comparing essentially to the double knockdown condition because we can't specifically deliver the molecule to only the tumor or only the microenvironment. And the significance that was observed for both CTIP2 and GFAP disappears in that double knockdown condition, suggesting to us, and this is our initial model, that PTPRZ1 is influencing migration and cell fate in a catalytically independent manner. So this brings us to our model, which is that PTPRZ1 may have a role in cell-cell communication. And we're really excited that we were able to come up on this through using our HOT system. We think that this is a proof of principle that the model can be used to look at cell-cell communication and really to study that context. At baseline, we do think that PTPRZ1 itself has a role in stemness and migration, but when it's communicating with the peritumor cells, this upregulation of PTPRZ1 maybe has a bit of an antagonistic role. This is particularly interesting because after some of the earlier papers in the last 5 to 7 years on PTPRZ1, I think that it's been kind of an attractive target because it's something that was upregulated in the tumor, but not necessarily expressed in the healthy brain. And I think that this kind of gives us pause as to how good of a target is this? Because if we do inhibit PTPRZ1 or go after it with some sort of small molecule, we may have that same role on the peritumor cells. So will the effect on the tumor outweigh that, and would that be a positive? Or could it perhaps even make the tumor worse, which would not be what we're looking for? Anyway, these are all important discussion points, and I think that there's a lot to follow up on in terms of understanding the mechanism for how this is happening, which could really give us more insight into what's going on there. So in conclusion, HOT can be used to model human GBM and maintain cell type heterogeneity and fidelity. And we're really excited about using this system to think about tumor environmental interactions. From those experiments, we saw that PTPRZ1 is involved in tumor environmental communication and that we identified this opposing role in migration and cell fate. So I'd like to just take a second to acknowledge the lab, a really amazing group of people and really amazing collaborators, especially David Nathanson and Kunal Patel, who have contributed to this study, as well as a variety of funding sources, and especially the patients who donate tissue through the Brain Tumor Translational Resource here at UCLA, as well as a lot of collaborations and good insight from the Brain Spore here at UCLA. And I didn't put it on the slides, but I'm sorry for not highlighting it. This study was recently posted to bioRxiv, so if you're interested in more and want to check it out, please do. Thank you all for your attention, and I'm happy to take questions, and I will get out of this so I can see you now.
All right, thanks so much for the talk. I'll just start us off, and whenever anyone else has questions, feel free to unmute and chime in. Yeah, I was curious about the organoid as a model system in terms of the sort of spatial aspects of it, right? Maybe two things to start that I thought of. First, you have this sort of organoid outside interface, so I was kind of curious if that has any physical analog or biological analog. And then of course we're talking about invasion and things like that, and the brain has a pretty unique environment there. So are the organoids a useful model for exploring like stiffness and sort of that aspect of migration and invasion?
Let me answer your second question first about some of the physical properties. I think that this is an open question. I'm not sure that we know that much about this. I know that there are very important biophysical properties that exist in the brain and that these are highly dynamic during development as well. So I think that some of that could be benchmarked even further. However, I think that based on some of the phenotypes we're seeing, we think that some of those properties might be recapitulated in the system. Your first question, I'm not sure I understood it, so if you could repeat.
Yeah, so I was just curious about the sort of biological, like, do you consider the organoid, media interface to be equivalent to, I don't know, some like it. Like, are there differences between like inside the organoid? We're pretty confident about the sort of fidelity, right? But like at the other edge effects in terms of nutrient access, like these guys exchange waste much quicker, right? And does that alter their expression relative to the core of the organoid?
That's a great question. I don't think we've looked at that sufficiently, and I think that that's something that we probably should look at. I think that we were surprised to see that there are some differences in how the tumor responds to certain conditions with essentially what I'm considering to be almost a buffering of the organoid. And I think that there's interesting mechanisms for how that's happening, but some of these questions about like waste removal and what are the differences between the core and the outside. We know that the core is more hypoxic because there are just less nutrients that get in there. We know that there's some like differences in nutrient availability, especially as the organoid gets older, but we haven't fully characterized that in terms of what it means for the tumor and what it means for kind of the fidelity to the patient context. So I think those are really excellent points that we should think about.
And I could do another one. Sort of in the same vein, right? Like you showed us those really interesting rosettes. So I was kind of curious if there was any sort of generalizable spatial biology around the rosette, right? Like, is there a significance to being, are the rosettes polarized in any way? Is there significance to being more in the center, like the core of it, versus the outside, or having a certain distance from the rosette? Does that affect the biology? Those cells?
So we haven't looked at that too much in terms of the tumor because by the time we do the transplantations of the tumor, a lot of those rosettes have disappeared. But in terms of normal development, I think that there's really interesting biology around those. So the radial glia cells do have this kind of polarity to them where they have one process that goes down to the ventricle and then another that goes up to the pia during development, and that is imbued by a polarity. And so that's why the rosettes form. And I think that for a lot of like neurodevelopmental or neuropsychiatric or patient-derived cell studies, changes in that have a lot of meaning in terms of, what's the density of the rosettes, what is the size of the rosettes, what is the timing of when that emerges. So I think that there's interesting questions there. We haven't explored that as much in terms of the tumor context, just because by the time we do the transplantations, those rosettes have mostly matured away, and so they're not there anymore.
I was curious about the your sort of meta-analysis tools, right? I think you mostly talked about focusing on the tumor, but is that a sort of a generalizable tool to a more like a more heterogeneous input data set? Because we often have these problems where we have spatial omics data that can be spotty, or we're just not entirely trusting of the batch effects of working with, say, FFPE tissue.
Sure.
So,
Yeah, no, I'm hoping that it will be. I think that that's really one of the main goals that we've had for why we developed that is I think that there are a lot of applications, whether it's different tissue systems, whether it's different data types, where this type of heterogeneity and batch effect can be confounding, but where having kind of a uniform analysis of what's going on can really empower more usage of existing data and integration of data sets. And so the tool is on GitHub. Feel free to check it out. And if there are any modifications that need to be made to be specifically usable for more spatial style data sets, we'd be happy to brainstorm with you and make modifications accordingly. We really do want that to be something that can benefit the community. And we've been really excited about the quality of data that we've gotten from it up until this point.
And I was kind of curious about the inverse problem, right? So it seems like you're using the tool to kind of figure out like what's at the core of like what's the common fiber connecting all these different samples. But when we work with these large data sets with hundreds of patients, say like sometimes we're interested in sort of what makes each patient different, right? So could you sort of flip this thing and use it to like filter out the sort of fundamental GBM-ness and then find out like this unique GBM-ness of each patient?
Yes, 100%. And we've been doing a bunch of that. It just every time we present it to people, they always want to know what's the same, not what's different. And so that's how we've kind of started telling that story. But that's the beauty of it. And that's part of why we wanted to do it. So absolutely, you could ask those questions with it.
And do you see sort of a clinical effects of sort of these molecular uniqueness traits, or I mean, I feel that's much harder, but you must have access to the clinical metadata, right?
Yeah, I don't think that we are powered in this study to do that yet, because for some of the samples that we put into the meta-atlas, we don't have patient information because it's coming from published data sets. But as kind of UCLA's repository of this accumulates, I think that we really are getting across labs and across projects many different single cell studies. And over time, we will be able to make some of those predictions. And so I think that it could do that. I just don't think we're powered to do it quite yet. Great.
All right. Well, if there's no more questions, I'll just thank you one last time for a great talk. And for everybody else, this is the last spatial biology seminar for a while. So I guess stay tuned. We'll see what happens. But yeah, thanks, Aparna.
Thank you so much. It was great to see you. Take care.
Bye.
Bye.
Thank you.